Hello everyone, good afternoon. I'm Lakshmi Gautambusa. Today I'll be doing presentation on topic identification of virulence genes in Pyrenophora teres F teres associated with the barley genotypes by using QTL analysis and seedling assay. In this presentation, I will be talking about introduction, impact of disease on barley production, importance of research, research objective, methodology, expected outcomes, and potential contribution and conclusion. Now let's talk about the introduction. As you all know that barley is the second most important crop in Australia after wheat in terms of grain production, and it is grown across a wide region from southern Queensland to Western Australia. While talking about the Australian barley production, over 8.6 million tons of barley is produced annually in Australia, which covers around 3.9 million hectares of land, and it has huge economic value, which contributes around 2.2 billion Australian dollar to the Australian economy. Now let's talk about the problem caused by net blotch diseases on barley. There are several biotic and abiotic factors that causes direct impact on crop yield and grain quality. Among all the biotic factors, diseases in barley can directly impact on crop yield and grain quality. The estimated average annual loss in Australian barley industry due to diseases is around 252 million. And then the net loss caused by Pyrenophora teres causes most significant loss in barley production. The estimated annual loss caused by net blotch diseases in barley is around 62 million Australian dollar. While talking about the net form of net blotch, it alone can lead to a loss of around $21 million per year, which means a lot to the barley industry. Due to the impact caused by net blotch disease on barley industry, there is urgent need for doing research for effective and efficient management of diseases. This research will help in identification of virulence genes associated with Pyrenophora teres F teres, which is very crucial for disease management. And this research will help in addressing the net blotch disease, which can potentially reduce losses in barley production and provide economic benefit to the Australian barley industry. This research will also help in enhancing the understanding of host pathogen interaction that will help in breeding program for the development of resistant barley cultivars, and it will enhance sustainable net blotch disease management in the future, which is very crucial. Now let's talk about the project aim and scope. The general objective of this research is to identify virulence genes associated with Pyrenophora teres F teres in barley genotypes using QTL analysis and seedling assay. And this research has a broad scope with substantial potential for this impact, and this will contribute to the development of effective strategies for controlling barley net blotch disease and for the identification of resistant barley cultivars, and helps in understanding of molecular mechanism underlying Pyrenophora teres virulence and disease development. This will lead to the more effective disease management practices and contribute to the breeding of resistant barley cultivars, informs targeted interventions, and helps in promotion of sustainable agriculture practices. Now let's talk about originality and motivation towards this research. There were previous several studies that have provided limited understanding of virulence genes in Pyrenophora teres and F teres. Therefore, there is an urgent need to manage barley net blotch disease and develop resistant barley cultivars for effective disease management. So, this research is motivated towards integration of QTL mapping and seedling assay that is used for genetic identification, and this research is motivated towards contributing to sustainable agriculture practices and reducing economic losses caused by barley net blotch disease. In addition, this research is motivated in bridging the gap between molecular genetics and phenotypic evaluation that provide interaction between virulence genes and barley genotype. Overall, this research is motivated towards understanding about the genetic basis of barley net blotch disease. Now let's talk about research design. Culturing of the Pyrenophora teres F teres is very important for beginning of the research. After that, we'll select 4 Australian barley cultivars: Escape, Pyrer, Beecher, and Commander with varying level of resistance. And then we'll use 190 pathogen isolates for phenotypic evaluation. The experimental design is based on split plot design. We'll be having 3 replication. 4 seeds of 4 barley genotypes is shown in each pot, and they will be grown in under the control condition. After that, we'll be doing phenotypic evaluation based on seedling assay to identify disease severity and response of different barley genotypes to the pathogen, as well as to investigate the pathotype variation and host resistance in the pathogen using phenotypic techniques. Now let's talk about seedling assay procedure. That is how we carried seedling assay. First of all, we prepared conidial suspension by culturing 10 mycelial pog on V8 media plates, and we incubated V8 plates in specific condition, that is 24 hours in the dark followed by 24 hours in UV chamber and again 24 hours in the dark. Then we have collected conidial suspension using distilled water with 220. And we quantified conidia using hemocytometer, and we adjusted the required volume of conidial suspension. And inoculation of conidial suspension to healthy plants are done using 3 ml per pot using Ojito airbrush and meeting compressor kit for inoculum. After finishing inoculum, we place those pots in a 95% humidity chamber for 24 hours and then transport to a controlled environment to check the host response. After 9 days, we evaluate the size and form of lesions, chlorosis and necrosis on the second leaf using a scoring scale developed by Teckhouse. Then we categorized isolate as a virulent and virulent based on the scoring. The score between 1 and 5 was scored as a virulent isolates, whereas the score between 6 and 10 is categorized as virulent isolates. Then we will be doing genotypic evaluation using DART sequencing and QTL mapping to identify genetic variation and markers associated with virulence genes that helps to determine sequence variation contributing to the pathogenicity. And for genotypic evaluation, we culture parental isolate stored at 80 degrees centigrade minus 80 degrees centigrade on half strength PDA medium at 22 degrees centigrade for 10 days, and we scrape the scospores from culture isolate. Then it is sent for DART sequence analysis and QTL mapping. The DART sequence analysis helps in the identification of genetic variation and markers associated with virulence genes. However, the QTL mapping will identify and locate the genomic regions associated with the genetic basis of disease resistance in barley genotypes. Identifying QTLs also helps in understanding the genetic factors contributing to the resistance against Pyrenophora teres, and data will be analyzed and interpreted using different softwares and bioinformatic tools such as DART software package, statistical analysis, etc. While talking about the expected outcomes, this research is aimed at identification of virulence genes or QTLs associated with disease severity and resistance or susceptibility in the barley genotypes. This research will enhance understanding of molecular mechanism in Pyrenophora teres, and the outcomes will improve knowledge of genetic basis of virulence and host pathogen interactions. This will help in development of net plus resistance variety that will implement targeted control measures based on identified virulence genes, promoting sustainable agriculture practices and ensuring long-term sustainability of the barley production. In conclusion, this research study aimed to investigate the genetic basis of virulence in Pyrenophora teres and its interaction with barley genotypes. The identification of specific virulence zinc and genomic region associated with disease severity through integration of QTL mapping, seedling assay, and genotyping techniques, we can gain valuable insight into the molecular mechanism underlying pathogenicity. These findings will promote sustainable agriculture practices and ensure long-term sustainability of production by effectively combating net blotch diseases. Here are the references that I have cited, and I want to express my gratitude to the researchers and authors whose works has greatly influenced this presentation, and their valuable contribution have helped me to shape and support my findings and conclusion. Last but not the least, I'd like to express my sincere gratitude to my supervisor, Dr. Enke Martin, co-supervisor, Dr. Buddhika Amarsinge, for their continuous guidance, support, and valuable insight throughout the research process. And I'm very grateful to the University of Southern Queensland for providing me the necessary resources and facilities for conducting this research. Thank you so much all for your attention, and if you have any queries or questions regarding this presentation, please feel free to contact me and post it into the forums. I'm happy to give the response.